Okay, before we get started on lecture, let's just acknowledge the stress that is occurring as you are preparing for your first lab practical. And then I want you guys to have the opportunity to ask questions if you need to, and for me to tell you what has been successful for many people in the past. So first off, expectations. This is probably something that you know, but just to make sure. Your expectation for your lab practical, it is not going to be multiple choice, right? It's going to be all written, and there will be no word bank, which means that you need to get prepared for that kind of thing. Now, I know some of you guys have been quizzed that way all along, right? You've been quizzed practical with no word bank and multiple choice. That tends to make you relatively well prepared. Some of the rest of you guys have been quizzed online with multiple choice questions. Realize that makes your quizzes easier, it makes your practicals harder. So I've tried to corral and make them all do the same thing, but I have failed thus far. However, I have been successful in that you should have a pretty standard expectation for what's going to be on the lab practical, and your lab instructor can give that to you. There is an expected number of anatomical terms questions. There's an expected number of histology questions. There's an expected number of axial, appendicular, axial muscle, appendicular muscle. There are a minimum number of cadaver questions, but there's no maximum, okay? Because models are easier. And we got to the point that like some people are only testing off models, and we're like, you can't only test off models, that's not cool. So there's no maximum, and there's a minimum number of attachments or actions that they have to ask, and that is all available to you if you just ask your lab instructor. They've got the complete write-up of what to expect. So they don't have to hold to it, but like if it says 14 and they did 13, nobody's going to be pissed off, you know. But if it said 14 and they did 6, right? So what they have to do is they have to submit their lab practicals for approval. Now, I also want you guys to know that you're not all going to have the same lab practical, right? Because in between lab practicals, all we have to do is move the pins, right? Change out the histology slides. So they're not all the same, even though there's like a standardized expectation. Does that make sense to you? Okay, do you have any questions that weren't addressed on lab practicals before? Okay, so here's something that starts happening the week before both lab practicals is that people stop attending lecture because they get stressed. I will tell you that when on Thursday we will start doing brain as the hardest thing to learn on your own. So you make your own choices. You are a grown-ass person. However, that would probably be an inappropriate choice, especially since you're having a quiz on Thursday. So making it more likely that you will come. So you're going to have a quiz on Thursday. We'll talk about what it's going to be about. It's going to be over muscles to reinforce the stuff that you're doing in lab and see how well you're doing it. Only I'm going to ask it from a different angle. Instead of identify that muscle all the time, I will tell you attachment points. You tell me the muscle. I'll tell you one attachment point and one action. You tell me the muscle. I will tell you something about it. I will go at it the other way from the lab, but with the same muscles, right? So that it can reinforce what you're doing in lab. That makes sense? And that muscle quiz will have no microscopic muscle stuff on it, just stuff that reinforces what you're going to be tested on in lab. And then it will have the set of notes that we finished today, okay? Which is nervous system tissue. And the reason I'm testing you guys on that is because otherwise you will not study it until after your lab practical is over, and it will be too late to get that terminology because the material gets harder after that. So you're going to do that first set of nervous system stuff, get that out of the way, and then I will help you reinforce muscles. So I'm trying to like support you dividing your time between lecture and lab. A lot of times they reinforce each other, but not at every single moment. And I'm just going to remind you one more time that when it comes to pitting them against one another, it's 80-20, right? Lecture's 80, lab's 20. But I want you to do well in both, really. Okay. Anybody have questions or clarifications they need? The last, I put this on SLCC Anatomy, but I just want to be really clear. The last open labs end Thursday. And there are no open labs from Friday until the following Thursday. once lab practicals start, open labs don't exist anymore. So just make sure that you're planning your time. Apparently the Saturday open lab was freaking chaos. It was like 60 people there. Yeah.
There were a ton of lab assistants.
I know, they just volunteered too.
Some labs where there's one lab assistant and she's just like pulling her hair out for some questions. So
yeah.
It was good. There was like one per cadaver, which was really nice.
That's really nice. I'll have to like send them a thank you because they just did that out of the kindness of their hearts on a Saturday. I have the best TAs. They are the nicest people. Okay, so we are going to finish this set of notes today. We'll briefly start on the other one. We're not doing anything hard about the brain today. We're just getting the basic protections of the brain. Okay, so what we were doing last time is we finished with talking about structural categories of neurons, and today we're going to look in depth at a multipolar neuron. So can somebody tell me, describe to me what I mean when I say multipolar neuron? What is it? Go ahead. Yeah, it's got more than 2 branches, right? So more than two projections sticking off of it. And so the reason we're going to look at this one in a little bit more detail is, of course, it's the most common type of neuron there is. And looking at it allows you to understand a lot of the structure and sort of suggests the physiology. Okay, so this, of course, is a multipolar neuron. So let's just start looking at the basics of it. Now, if you have a histology slide, I acknowledge that you can't really tell the difference between an axon and a dendrite dependently, right? Unless, of course, they stained it so you could see myelination, but they didn't in this one. So you would have to have it labeled or indicated in some way. But dendrites, they're typically referred to as receiving information, but the reason they say that is because they have a bunch of cell membrane receptors on them, right? And so they are actually receiving stimuli from another cell, a lot of times in the form of chemicals like neurotransmitters. However, the dendrites can actually respond to touch, temperature, pressure, pain, light, those things as well. But regardless, the information is coming in via dendrites. And then it is going to move through this, which is the soma or cell body, yeah, to this special region right here. I know it doesn't look special. It's called the axon hillock. What the axon hillock can do is it can take whatever kind of stimulus was put in and convert it into an electrical stimulus called an action potential, okay? And then it sends the action potential 250 miles an hour down the axon, unidirectionally. That means that, of course, by looking at any neuron, especially if it's a multipolar one, you can immediately tell the direction of information flow, right? Top to bottom on this one, right? Okay, that becomes really useful when you're in phys. You can look at the setup and see some of the functionality. Okay, we'll talk about the myelination in just a second, but before we do that, the axons, they can be really long. Dendrites aren't usually incredibly long, but axons can be really long. So the place they start is the axon hillock. But let me remind you what a nerve looks like so that you can put this together with a picture of a nerve. So remember that a nerve, of course, doesn't generally have any cell bodies in it, right? It's just bundled wires, yes? Okay. So that axon right there, imagine that it was attached to the neuron that we just saw, right? Okay. So this axon is running through a nerve, and there are so many axons in a nerve that sometimes they call an axon a nerve fiber. Does that make sense? I won't use that terminology, but you will see it used in the future. Okay? I won't try to trick you with it. Now, this is where it gets really important that you can distinguish between a neuron, a nerve fiber, and axon, right? So you have to get to the point that you're comfortable using that terminology. So the quiz that I give you on Thursday will assess whether you're really comfortable using the terminology correctly, right? Okay, so that's sort of the relationship between an axon. And notice that this axon up there at the top has a myelin sheath on it, and we'll get to the function or the structure of a myelin sheath in just a second. Okay, I forgot to say this. Let me tell you just a little bit about the soma or cell body before we leave. Remember that these cells have like these owl-eye nuclei, right? Which is like a nucleus with a nucleolus in the middle. And then generally speaking, when you look at a neuron, it's going to like have a granular cytoplasmic appearance, and that's because There's a whole bunch of rough ER in there because most neurotransmitters are amino acid based. So it's going to have a decent amount of protein synthesis. There's usually a whole bunch of mitochondria in there as well because they're really energetically expensive. Okay? Okay, so let's look at this axon and talk about them being really long. So we will see that almost all of your neuron cell bodies are sequestered within the central nervous system, but of course the wiring is not sequestered. It can't be. So you can have an axon that's incredibly long. So if we go back to this picture that we looked at last time, the neuron cell body that is actually going to control the contraction of my tibialis anterior, right, is going to be, the neuron cell body is up here in the lumbar region, my spinal cord, okay? And the only thing that runs down there is an axon, yeah? They're really long. It's one axon running down there, okay? So it's a pretty, I mean, it's not that impressive in me, right? I'm short. It's more impressive in him. It'd be way the hell more impressive in Shaquille O'Neal if he was here, right? So they're really, really long, those axons. Okay, so let's talk about what axons can do and a little bit about the concept of neuroplasticity. Okay, so this is an oversimplification, and I will make it truer when we get to the next set of notes. But I want to imagine that I'm a packet of knowledge and you are a packet of knowledge, and I can physically connect those two packets of knowledge by making what we call synapse, and we'll talk about that in just a second. So what I do is I actually get very, very close to her to the point that I can actually release chemicals to communicate with her. That makes sense? Okay. And the chemicals will be released from here. Yeah, we'll talk about the details of that in just a second. But what I can do is I can also make that packet of knowledge connect to other packets of knowledge. Yes? And I can do that because there is literally no limit to the number of times that an axon can branch. Yes? And so I want to imagine, and again, this is metaphorical, a packet of knowledge is not a neuron, right? It's a collection of neurons. So I want to imagine that I learned something in 1610, right? And then I am going to connect it with something that I learned in anatomy, and then it is the same packet, and I'm going to connect it with something that I learned in physiology, right? And I have the capacity to make new neural networks, which is what is happening in your brain, large scale, when you are learning new things. Does that make sense to you? And so you are actually changing the physical structure of your brain when you are learning new things, because there is no limitation to the number of times that this can branch. In addition, what if I just wanted to reinforce this packet of knowledge, right? If my knowledge, instead of going broader, it went deeper. What can happen is, if I make one connection to you, then our communication is pretty quick. But what if I make 25 or 30? Then I've reinforced the connection, and it's really, really quick, right? So this is one of the concepts of neuroplasticity. It says that you can change your brain based on what you can do. And that is one of the most fundamentally hopeful things I've ever heard, honestly, with me. And it's not even not true. It was just a little bit of a simplification because it's neural networks and not just single neurons. Okay, so now let's talk about this concept when you get to the end. So when an axon branches, it's called a collateral. This one's just showing one. You only ever have one axon hillock, but there's no limit to axon collaterals. And then all of them will branch when they hit the end. So I want you to imagine that this is the soma, this is the axon, yeah? And then at the end, you will always have branches at the end. These are called axon telodendria. Telo meaning end, dendro meaning fingers. So literally the fingers at the end, okay? But notice that the fingers don't look like mine, they look like ET fingers, yeah? And then I realized that my references are getting stale because you haven't seen ET, but you should. Okay, so they look like little ET fingers. And so what happens is at the very end, they're going to be bulbous at the end because what is stored there? Yeah, that's where you store and release neurotransmitters. So those are called, sorry, nobody can agree what to call them. They're either called axon terminals or synaptic knobs or terminal boutons or synaptic whatevers. It doesn't matter. They're all the same damn thing, and nobody can decide what to call them. The place that neurotransmitters are stored is what's actually important there. Now, what's the big deal about neurotransmitters? Neurotransmitters are how this one will communicate with the next one. They will allow me to, for instance, when I get a stimulus from here, right? So these are my dendrites. I get a stimulus. It transmits through me, and then I splash it onto you. If you're the next neuron, then you're starting the whole thing over again, right? She's being splashed with chemicals, right? She can receive them, start it, send it on. Does that make sense? Okay, so synaptic knobs are synaptic terminals, and I'll talk about axons again in just a second. Okay, so let's talk about the myelin sheath and what the hell's going on with the myelin sheath. So axons, some of them, but not all of them, are myelinated. And myelinated looks like these sausage links that you're seeing on the axons, but not the dendrites, yes? Okay, so I'm going to give you a simplified version of this. It's a little bit more complicated than this. It's a lot more complicated than this, but basically what I want you to imagine is that this is insulating the axon to both allow for transmission and speed up transmission. A metaphor is like if you're doing a science experiment with your kid, or if you are a kid doing a science experiment, and you need the freaking potato battery to light up your light bulb, right? You got a copper wire. It's only that long. Do you need to insulate it? No, you don't. But now if you want to run it through a Rube Goldberg machine, right, because you're a smart kid, you ain't just making a potato battery, right? It's going to knock over some dominoes and swing a whirligig. Then what happens is if it's running by things, the longer it gets, the more you need to insulate it. In the experiment, it's because you're going to burn the damn house down. You won't burn your house down, but what happens is if you send an electrical signal up against something, you can lose the charge, right? So the longer the axon, the more need for myelination. If I have two neurons right next to one another, it's unnecessary to myelinate between them. But if we look again at two situations, first, I don't need to myelinate these two neurons that are right next to one another, but if this one wants to communicate with his friend over here, I need myelination. Yeah? Wherever you have myelin, it tends to make the tissue white. Go ahead.
Does that have an effect on neuromuscular disorders and like where you lose kind of function first?
Yes. Mm-hmm. So, and we'll talk about MS with that relationship in just a minute. So hold on to that question for just a second. Okay, so myelination it does need to occur in the brain when the parts are separated. So if I'm communicating between the brainstem and the prefrontal cortex, you definitely need myelination, or between the two halves of the brain. But if the two cells are right next to one another, it's not necessary. But of course, if you're doing peripheral nervous system, everything is really far apart, right? Because it's just wiring. So what is this? The nerve is bundle after bundle after bundle of myelinated axons, right? Also protected with connective tissue, right? So you will see that within the brain, the wiring is going to be white, yes? It's called tracts or pathways. Within the peripheral nervous system, almost every damn thing is white because it's almost all myelinated axons. Does that make sense? So when you are looking at the cadavers and you see a white thing, it's probably a nerve. Yeah? Okay, they're not actually bright white. They're like kind of funky, dirty white, but whitish yellow. Okay, so let's talk about how myelination occurs briefly, and then we'll get into a little bit more details of it in just a second. So there's two ways to myelinate. Here's the first way. The first way will end up looking like this. Okay, notice you'll end up looking like sausage links. So the way that occurs is this is an axon. The yellow thing is an axon. What you're going to do is you're going to take a certain type of glial cell, and the glial cell is called a neurolemocyte or a Schwann cell. And it's going to devote its entire developmental self to wrapping around and making a sausage, right? And it's going to wrap and wrap and wrap to the point that it pushes out almost all of the cytoplasm and it pushes aside almost all of the organelles, right? So basically what you have is it's devoting its entire self to making a little sausage. And since the cell membrane, which is mostly what you got on there, is primarily made of phospholipids, right? And it's really like a fatty insulating sheath, right? It's a fatty insulating sheath. It's mostly fat, okay? So why is it white? Well, because if you have a single layer of saran wrap, it tends to be transparent, but if you have a roll, it tends to be opaque, right? So it tends to make the tissue look white. Does that make sense? Okay. So this is how you do it in the peripheral nervous system. There's one other way to do it. It accomplishes the same thing. It will be clinically a little different, though. So, go ahead.
Is the endoneurium also in there wrapped up? Or?
So, the myelin sheath
Yeah.
is deep to the endoneurium.
Okay. Okay.
So, you've got endoneurium, myelin sheath, right? And then the cell membrane. Okay, that's a good question. Okay, so how do you myelinate in a nerve, which is peripheral nervous system, right? Like my radial nerve running down my arm. You myelinate it with that thing that you just saw. These cells are called neurolemocytes or Schwann cells. You guys, unfortunately, are in the decade in which you are going to have to learn both eponyms, which is like when something was named after a proper name, and the new anatomical term because we're trying to switch. So they don't want you to learn Schwann cells or nodes of Ranvier anymore. But the problem is, if you don't learn them, you won't be able to talk to the people who are older than you. So, right? So you actually want to be able to know them, right? You want to know what the surgeon means when he says nodes of Ranvier, right? So you have to learn both. And I did too, just so you know. They changed them after me, so I have to teach you guys the right way and remember not to say it the wrong way all the time. Okay, so these are neurolemocytes of Schwann cells, and that's how it's done in the PNS. In the CNS, it's done that way. Notice it accomplishes the same thing, but the cell itself is sitting off to the side. These are called oligodendrocytes, which means oligo, many, dendro, fingers, site cell. It's a many-fingered cell, so it wraps around. Okay, why the hell do you care? The reason you care is because if you damage this in the PNS, these will try super hard to help you with regeneration. If you damage this, these are like, I'm out. And you hardly ever get any white matter regeneration in the central nervous system because of that. And we'll talk about nerve damage and regeneration at the end of this set of notes. But basically, never damage your nervous tissue, but if you damage it, damage PNS, not CNS, right?
Thanks,
I mean, you don't get to pick, right? Huh? What?
I'm sure you do that, yeah.
Just if you had to choose. You don't generally get to choose. Okay. All right, so oligodendrocytes. So let's talk briefly about these little areas. These little areas right here where it's not myelinated, like the rope on your sausage links. These are called nodes. Again, they can't decide what to call them. They used to be called nodes of Ranvier because he found them. And now they're called neurofibril nodes. But if you see node, that's what they're talking about, so it doesn't really matter, right? That makes sense? Go ahead.
So then if you tested us on that with just like a short answer question, if we wrote one or the other?
I don't care.
Okay.
Yeah. And actually node is fine because node, there's only one node there, right? So it's the only node that's there. So I don't try to nitpick you with like eponyms and that kind of stuff. I want you to know it well enough that you will be able to recognize it and use it in the future. If I write stuff that's really specific about terminology, it should be meaningful. It should be terminology that if you got it wrong, you would have a misunderstanding of the concept. Okay, so those are your nodes. So just based on what you know right now, you should very easily be able to look at it, any neuron that's drawn and tell direction of information flow, correct? Axons versus dendrites. Nodes. Myelin sheath, axon hillock, soma, telodendria, synaptic knobs. And then you should also be able to tell me whether this axon is running through the CNS or the PNS. Yeah, it's PNS because that's the way that it's myelinated, right? So the good part about the discomfort of learning nervous system anatomy is that it makes nervous system physiology make sense more quickly, right? You can look at it and immediately have some assumptions that you can confirm, right? Okay, so, all right, so now let's talk briefly about synapses. Okay, so what is a synapse? A synapse is when this thing gets to the end, what is it going to do next? What it's going to do next is it is going to communicate with the next cell. And so now, because you know about axons and dendrites and direction of information flow, you can put any two neurons together, and as long as you understand the structure of them, you can tell which one's presynaptic and which one's postsynaptic, correct? Yeah? presynaptic, postsynaptic neurons. The site of communication is going to be called the synapse. There are multiple subtypes, but we'll worry about that at some other point. So you can tell which direction information is flowing. The synapse, they look like they're stuck together, but they're not. They're microscopically separated. So if I blew up one of these guys and looked at the site of communication, what you would see is, so here's the axon, here's the little ET finger, right? The synaptic knob or terminal. I will not test you on this physiology yet. It's a preview of the physiology that you will get in future semesters. But I'm just going to tell it so that you can see why understanding what's going to go on here is relevant. What's going to happen is when you send that electrical stimulus that started at the axon hillock and it goes all the way to the end here, right? What will happen is it will cause exocytosis of whatever neurotransmitter was stored there. And the exocytosis of the neurotransmitter will then communicate with the postsynaptic cell. And the postsynaptic cell, you are going to dump neurotransmitter onto the postsynaptic cell, receive it generally with the dendrites on the postsynaptic cell, start the whole damn thing over, right? That makes sense? But just so you know, you don't always start another one. Sometimes in your brain, it's just as important for me to tell you to start and you to stop, right? So you can send signals to do either. I could send a signal to you that says start, depends on what receptors it binds to. But you could hear that signal as stop, because you do not want everything on in your brain at the same time. You will be batshit crazy, right? You ever had one of those days when it feels like everything's going on at the same time? You can't get anything done. You're damn near about to pee your pants. You're like, I don't even know. I can't, no. You have those days, right? It's not just me. Okay, all right. So many days. Okay, so it's called a synapse. This is Melanie's very favorite part of physiology. I go bonkers on it. But you don't have to do it in anatomy. Okay, so you should be able to now, by looking at these two, be able to tell which one's presynaptic and postsynaptic, right? Label two different axons, tell direction of information flow, right? Axon hillock, telodendria, synaptic knobs, dendrites, soma, myelin sheath. So you can tell a lot from just putting two together if you know the anatomy. Okay, now, last little tidbit about this is that neurons don't just communicate with neurons. Neurons can also communicate with muscles or glands. So this neuron right here, the presynaptic one, could be communicating, for instance, with a skeletal muscle fiber and telling it what to do, or smooth muscle or a gland. When it's neuron to neuron, they usually call it a synapse. When it's neuron to muscle, they call it a neuromuscular junction. And when it's neuron to gland, they call it a neuroglandular junction. Good? But we won't deal with those much. We'll deal with them in phys. Okay, so that's quick and dirty about neurons. I was focusing on multipolar neurons. Now I just want to talk about the unsung heroes of the nervous system, and that is your glial cells. So if we go back and look here, everything in this picture that is not neuron, all of these little cell-looking things are glial cells. Mostly what stayed there was just the nuclei of them, so they don't all exactly look the same. And I think I told you guys this before, but when they named them, they thought they were primarily just space fillers because they knew that the neurons were important, right? The neurons were the star of the show. But they were like, these are just glue. So they named them literally nerve glue, neuroglia. And as far as the proportions in the brain, they are about 50 times more numerous, but they're so much smaller that they end up making about half the brain volume. Okay? Now, functionally, they're pretty damn important, because when we started figuring out quite a few neurological disorders, we figured out that although what happens is the neuron starts behaving badly and you have a neurological disorder, a lot of times the disorder does not originate in the neuron. So let's talk about these. So let's talk about glial cells. You have 4 glial cells, 4 different kinds, in the CNS, and you have 2 in the PNS. So the metaphor that I always use for this is that neurons, during the process of development and differentiation, became so specialized to receive, transmit an electrical stimulus, and release neurotransmitters but they know how to do almost nothing else to maintain their neuronness. For instance, they don't know how to feed themselves properly, they don't know how to get waste out properly, they have no idea how to communicate effectively without something myelinating them, and they have no self-protection capacity. So think about the Olsen twins, right? Being raised as movie stars. Do you think they know which side the gas goes in? Or how much it costs? Or where to get their favorite latte? Or what to do if their cell phone gets cut off? No, they probably don't. Well, they're bigger now, so perhaps they do. But, so, you have this entire support staff. And the support staff is specialized. And when you are in LA, you get a bigger support staff than when you are on location in Africa. CNS, big support staff. PNS, small support staff, right? So let's look at the big support staff. The big support staff is here. These are the Olsen twins, right here. They don't know how to do anything for themselves. Sorry if you're really competent Olsen twins. I don't know. I don't have another twang word to use. Okay, so what's their support staff? These are the communication directors or publicists, right? In the absence of those oligodendrocytes, they can't get their messages in and out, right? The oligodendrocytes fail them. They can't communicate any further than the cell that's right next to them. Yes? Okay. Next, what we have is these guys. They're called astrocytes. I bet you can figure out why. Because they look like stars. Okay? They thought originally that those were primarily just like the scaffolding of the nervous system, because they're like holding on to one thing and the other thing. They got a blood vessel and an axon over here. And that's part of what they do, but not all of it. And they also tend to wrap their little feet around capillaries in the brain. And by doing that, they actually act as bouncers to make sure that nothing unsavory gets at the twins. They actually aid in maintaining the blood-brain barrier. And the blood-brain barrier, we'll talk about it more in the next set of notes. There's funky stuff that goes through your bloodstream. You don't want it to get into your central nervous system because your central nervous system is really bad at repairing itself. So what happens is these provide the last check on access to the central nervous system. Yeah? So they make sure that whatever funky stuff is in the capillary doesn't get through, but they also will allow, for instance, glucose right through. So they're really selective about what gets in. Then these guys right here account for the fact that if you can't get stuff out of the bloodstream very effectively, you also probably don't have white blood cells, the COPs, in your compound, right? So you got your own little ninjas, okay? And your own little ninjas are your microglial cells. Microglial cells look unimpressive, but they are freaking Jackie Chan, and they can whoop up on anything that gets near the twins. Does that make sense? So those are like the little security force that's in there. And then the last thing, and these are a little hard to sort of conceptualize, and I don't have a good metaphor for them, but these guys are, okay, so let's show you where they're coming from. Do you see that right here they're trying to show you that they got a chunk of tissue and they got a chunk of this space right here? Your brain is hollow. I don't know if you know this, but your brain and your spinal cord are both hollow. So this one has a chunk of the hollow. So let's look at what happens at the chunk of the hollow at the edge of it. These cells right here are lining the hollow, right? They're up against the brain tissue and lining the hollow. The hollow is called a ventricle. You'll learn those later. Okay, these are called ependymal cells because they look kind of like epithelial tissue, don't they? They're not epithelial tissue, they're specialized brain tissue, but they're called ependymal cells. And what they do is they selectively pull the best stuff from the capillary, like high levels of the ions you need for neurotransmission, calcium and sodium and potassium. And then they pull a whole bunch of glucose and then isotonic fluid to make sure it doesn't screw up the, make cells lyse or anything. And they're going to pull it from the capillary and dump it into the ventricles of the brain. And there it's cerebrospinal fluid. So those cells are responsible for making cerebrospinal fluid. And you've heard of cerebrospinal fluid, right? Because you've heard of spinal taps and that kind of stuff. Your brain is like got fluid on the inside, it's also floating in fluid on the outside. That make sense? Okay, so this is your support staff when you're in LA. When you go on location, you don't get to bring them. You only get to bring 2. And so if I am in the PNS, the glial cells, we already know one of them. because this glial cell is responsible for myelination of the TNS, and it's called the neurolemocyte. Yeah? And then this one, remember we don't have very many neuron cell bodies outside of the CNS, but when we do, they're in a little blob called the ganglion. Yeah? And each one of them is completely surrounded by a cell that tries to make sure it has the proper nutrients and gets rid of wastes and does as much protection as it can. It's a general assistant that you've got right there, and they're called satellite cells. Do you see why they're called that? Satellite cells, because they're sort of rotating right around. Yeah, exactly. Okay, so of course this cell is a little strange looking because of what structural category is it? That's a unipolar one. Yeah, unipolar ones are more likely to be found in ganglia than multipolar ones. So this structure right here is a ganglion right there, and it's myelinated by the neurolemocytes. Good? Okay, so as we'll come back to her question, does anything ever go wrong with these cells? Well, yeah. First off, although I've told you before that neurons are mostly amitotic, glial cells are not. Most of your glial cells have both the capacity and the propensity for regeneration. That means that, if you had to guess, which would be a more likely source of central nervous system cancer, a neuroma or a glioma? Yeah, gliomas are more common than neuromas. Neuromas do occur because there are a few mitotic cells in your central nervous system. But you can even be more specific. You can have a microglioma, you can have an astrocytoma, you can have an ependoma, you can have an oligodendroma. Does that make sense? Okay, so because they are the mitotic tissues that still exist in the central nervous system, they're more likely to be the sources of cancer. And so now we'll address what happens with demyelination. So there's also a disorder in which you demyelinate. So it's generally going to occur. Okay, so MS, multiple sclerosis, is what occurs when you, usually this is how it occurs. You get exposed to a virus, and we usually don't know what virus it is. And the virus ramps up the sort of sentinel nature of your immune system just because that's what it does. And when it does that, sometimes your immune system makes antibodies that actually make mistakes. They are supposed to be looking just for viral antigens, and they make the mistake and they start to attack the cells that make up your myelin sheaths. And when they do that, you get loss of myelination and loss of the capacity to communicate, not necessarily between two cells that are right next to one another, but between things that are broadly separated. So you're more likely to, for instance, with MS, have loss of sensation and motor activity in the periphery first before you do centrally. Does that make sense? It can move up. And so you can lose control of motor and sensation in the periphery, which made me think, and I was wrong, that it must be a demyelination of neurolemocytes. But apparently it's a demyelination of the oligodendrocytes. Getting the signal all the way out to your arms is the problem, not so much from your central nervous system to your arms. Because even getting it from, for instance, the cerebral cortex where it starts right here, all the way out, it's still a pretty long myelination process. So I don't quite understand it yet, but it's mostly demyelination of oligodendrocytes and not demyelination of neurolemocytes. And like all autoimmune disorders, there isn't really a cure for it, but it can go into remission, right? Yeah, there's a bunch of different types, yeah. I wonder if they're all oligodendrocytes. I'll have to look it up at some point. I don't know. So yeah, I just thought that my hypothesis was correct, that it would be these, because of course it's usually peripheral stuff first.
Do you know what Guillain-Barré is?
I have looked it up, but I don't usually teach it. If you send me an email, I'll see if I can fit it into a lecture. I don't remember it well enough to teach it though. Okay, so that's MS. Last thing in this set of notes is to talk about what happens when you damage nervous tissue. Well, it depends on where you damage it and what you damage. So first thing is that with very few exceptions, we know that if you damage the neurons, not very many neurons have the capacity to regenerate. It certainly does not mean that you can't pass off the functionality that you lost by losing the neurons to another part of your brain. Your brain has tremendous capacity for doing that, but I'm talking about regaining the actual tissue, not passing off the functionality. So mostly, with a few exceptions, if you damage the neurons themselves, you don't get them back, okay, whether it's brain or spinal cord. But what if you damage the white matter? Is there any difference between damaging the radial nerve, right here, and the radial nerve, you won't be at all surprised, is primarily responsible for sensory functionality on the lateral portion of your arm and motor functionality here, right, on the lateral portion of your arm. Is there any difference between the likelihood of regeneration if you damage that versus damaging the corpus callosum in your brain? And there is. Or damaging, for instance, the white matter of your spinal cord here. And there is. There's difference. So let's figure out why there's difference. So if you damage cell body, you're kind of screwed. In the PNS, however, if you damage an axon, you have the possibility, but never the guarantee, of regeneration. So let's look at how the possibility occurs. So the possibility occurs because if you have whatever kind of trauma, so first off, remember that a nerve is not a neuron, right? And if I damage my radial nerve, I could completely transect it, right? And damage all of the axons, and I would lose sensory and motor capacity. Or I could damage some of them, some sensory, some motor. So I could end up with anything initially from muscle weakness to muscle paralysis, from loss of partial sensation to complete loss of sensation, depending on how much I damage, right? So, but let's just focus on a single axon and try to see how you can predict whether a single axon is going to repair. So if you focus on a single axon, what happens is when you damage that axon, the axon degenerates because it's cut off from most of the cell organelles. There's still some mitochondria down there, but it's cut off from the nucleus, and so there's hardly any protein synthesis that's going to go on. It's like cutting off your arm. So it will degenerate. But as it degenerates, this region right here will start to try to heal and grow a new axon and grow new, ultimately, axon telodendria. And the cool thing is that these cells, the neurolemocytes, will try to help. And the way in which they try to help is they form a regeneration tube, and the regeneration tube is saying, hey, grow this way. And so I want you to imagine that this is skeletal muscle. And remember, if you damage innervation to skeletal muscle, it's like a teenager. It doesn't do a damn thing unless you tell it exactly what to do, right? So it's not doing anything at all right now, right? And now, what's going to happen is this regeneration tube is going to form. And the regeneration tube is more interesting than just anatomy because they figured out that the regeneration tube also releases chemicals that attract the growing axon. It's called chemotaxis. And they actually figured out what those chemicals are in the lab, and now they manipulate them, and they're trying to help with CNS regeneration. So it's doing chemotaxis. It's trying to say, come over here. This is where you need to go. And give it a little time. 1 to 5 millimeters per day is all it can grow and myelinate. And then potentially you could get innervation to the effector restored. That's one axon, right? So one axon might be restored if you're lucky. But now damage the damn whole nerve. And then realize that surgically you're probably going to go in and have to put it together. But what can the surgeon do? Well, it's mostly a gross surgery with a little bit of microscopy involved. So what they would do is they would, of course, put the epineurium together, right? And they'd try to stick the fascicles in the right place. You can't get every single one of those in the right place, right? So, and then it only grows 1 to 5 millimeters per day, which sounds really fast until you realize that if I damage it up here, it's got to grow all the way down here, right? And so it can be years before you will know how much reinnervation you will get. And that's a big deal because the other thing is that muscle tissue will degenerate and become scar tissue if you don't keep it alive. It's use it or lose it, which is why you have to do physical therapy, even if you don't ever know whether you're going to regain functionality. Muscle tissue is incredibly energetically expensive. If you are not going to use it, the body just says, you don't need that tissue anymore. We don't have that kind of money. Go ahead.
So if you damage a nerve, like right here, then I'm imagining like putting it back together with the old piece, but you're saying like you'd have to grow all the way back down.
Has to grow all the way
So
back
it
down.
couldn't just like find its little bit.
Well, the surgeon would, for instance, put it back together if they were broadly separated,
Yeah.
right? But then all of these, if it was completely severed, all of the distal injury will die,
Yeah.
right? You'll still get to keep the basic connective tissue, right? But you'll have to remake all of your myelin sheaths. You'll have to remake all of the axons. And so your responsible surgeon, responsible doctor will not tell you you are definitely going to regenerate this functionality.
Yeah.
They will say, wait and see. We are very hopeful.
Right.
2 years, right? They don't know. It's not them, being irresponsible. Okay.
So it can just find its way down there? Like, say it is like your sciatic nerve or, it can just find its way all the way down where it's supposed to go.
You can hope it finds its way down there, and sometimes it hits the wrong tube, which is pretty damn funny, right? And your nervous system rewires so that if I do something here, I will actually feel it over here. I don't feel it any place differently, but if it rewires to a different part of my brain,
So,
then I think I'm feeling it differently.
so
Yes?
it's like,
And you can control motor function differently. Also, it's pretty easy for your brain to retrain if I think this and do this, but all of a sudden it's doing that. Your brain can retrain, right? But the sensation is a little harder to get used to because you think something is happening here. Oh, there's a fly on my finger. No, it's on my thumb. Sometimes they just hit the wrong tubes, right? Yeah, it's pretty cool. I mean, not when it happens to you, but just theoretically. Go ahead.
Regeneration, does that only happen when it's like a physical trauma, or does that happen with a stroke as well?
Well, stroke is a physical trauma. It may not be a blow, but usually when people are talking about strokes, they are talking about losing gray matter, although strokes can lose white matter. And when you lose gray matter, the loss of tissue is mostly a permanent loss, especially if it's cerebral cortex loss. So, can you still regain functionality? Potentially. Better when you're young than when you're old. Better for women than for men as well, for reasons we'll figure out. Women actually have more redundancy on the functionality of the two hemispheres of their brain. So if you have a unilateral stroke, a lot of times this part already knows how to do part of it. But generally what's happening there is that I like to imagine the cerebral cortex functionality is just this sort of general, really powerful processing capacity. It doesn't really care what kind of information it processes, right? And so if you lose processing over here, left temporal lobe, and you lose speech capacity over here, if you can figure out that speech capacity needs to occur, you can pass it off to another processor and say, I just now need you to, instead of processing hearing, I need you to process speech. And they think, by the way, that that's one of the phenomenal capacities of the human cerebral cortex that makes us, arguably the most intelligent species on the planet. It's not so much what our brain can do, but the plasticity of what our brain could do, given a different stimulus, right? It is tremendously capable to take new information and process it in new ways, in a way that the cerebral cortex of lots of organisms is not. So with stroke, what usually happens is you pass off functionality. And it's not very quick to do it, but so we'll talk about strokes when we do brain next time too. Okay, what happens in the CNS if you damage something? If you damage something in the CNS, basically, so let's talk about this. Damaged gray matter, that's what we were just talking about. The regaining of functionality will, by the way, if you damage gray matter, what happens is usually the astrocytes move in and they just scaffold the place, and for all practical purposes, it's kind of scar tissue, right? They're in the place, but they're not doing the normal functionality. What if you damage white matter here? Well, it's interesting because oligodendrocytes, as soon as they get damaged, if you damage the white matter, they just say, I'm out. And then astrocytes move in, right? And they just basically, for all practical purposes, they form scar tissue impediment so that even if you could regenerate the axon, and you can't generally because the oligodendrocytes won't happen, you've got an impediment. So this is interesting because it causes problems with spinal cord regeneration and nervous system regeneration with white matter. We found some ways to work around this. One of them is, for instance, the spinal cord regeneration. We have figured out those chemotaxic chemicals that are used by neurolenocytes. And we can use them in vitro to try to get regeneration of CNS tissue. But as far as I know, last time I checked, those have not been used in vivo yet. They may have been used in animal models. I don't think they've been used in humans yet. We could check. But the other one is that we do have some capacity, but also some tremendous limitations for white matter and gray matter regeneration in the central nervous system, and that is embryonic stem cells. Embryonic stem cells are kind of an interesting thing. So an embryonic stem cell, if most of this research, by the way, got pulled from the United States when George Bush II removed all federal funding for embryonic stem cell research because he was very confused and thought that embryonic stem cells came from aborted fetuses. We will see why that can't be true when I explain to you what embryonic stem cells are. Okay, so what happens is at a certain stage of development, right, zygote, for instance, you have to be able to make every single cell in the body, right? You just have to. And then you go through multiple steps of differentiation. So let's say that I was infertile, and my family and I decided we wanted to do in vitro fertilization. It's really expensive. It's at least $10,000 a pop, and it's got about a 20% success rate, less as you get older. And so what they would do is they would give me drugs like Clomid to make me hyperovulate, right? And they would harvest the eggs, right? And then they would take somebody's sperm and do fertilization. And then they would let those divide up to about the 16-cell stage. We are still talking 16 cells, no differentiation, nothing recognizable as a human, okay? And then they put them in cold storage. And then they shoot them up with a turkey baster until as many as I want stick, right? That makes sense? It's expensive. You don't hyperovulate a single egg and then try it and then do it again. You usually do 20, 25 if you can get them, right? And you fertilize them and you freeze them. And even in Utah, ain't nobody want 25 children, okay? Okay? Once you figure out that you get less stuff if you have 10 or 12 children, because they get all your stuff, you think 2 is fine. What happens is those embryonic stem cells can make any cell in the body if they haven't differentiated yet, okay, at all. And so you keep them frozen. And then what happens to them after I've had my 10 or 12, right? Well, generally speaking, people couldn't decide what to do with them, so they just left them frozen. They're only viable for implantation for about 2.5 years. They're only viable for research for about 7 years. Those numbers may have changed a little bit if we got better preservation techniques. But generally, people just didn't decide. Or you could donate them for embryonic stem cell research, okay? And the embryonic stem cell research, they had to be tracked. There had to be a chain of custody that tracked them back to somebody using IVF, meaning that you couldn't make them just for that purpose, right? That's actually an international law. George Bush misunderstood. There's a lot of misinformation about how it's really easy to understand, because you understand it now, right? You're like, I don't know, but I wasn't president. I'm pretty sure I could have gotten this. So he pulled all of the federal funding from the National Institutes of Health and all of the other research organizations. So what happened is all of our scientists that did that left. So now if you get a spinal cord injury here in the United States, you do not have access to what they do, for instance, in Sweden. If you get a spinal cord injury in Sweden, you still have this narrow window. And it's still clinical trials, just so you know. We've made it work really well in rats because we can transect the rat's spinal cord in a rat and then stick the rat embryonic stem cells in there before any scar tissue occurs, right? But you don't usually get your spinal cord damaged in the hospital, right? You don't. So you've got to get the treatment occurring very, very quickly, and it's not occurring all over the place. So what has to happen is you have to get it in there before the astrocytes set in. And then when you get it in there, embryonic stem cells take their cues from the tissues around them as to what cells should be made, and they can actually make more gray matter and white matter. And with rats, when you can control all of the variables, the rat paraplegia studies in which they completely transected the spinal cord at the lumbar region and then injected rat embryonic stem cells, they got the first regaining motor and sensation in 2 weeks. And then they had regained almost complete motor and sensation by 6 weeks, and then by 2 years they were almost dead rats, but they had regained almost all of their functionality. But you can't have access to that kind of treatment here in the United States because all of our scientists left. And you can't really fly right after a spinal cord injury, right? So if you're gonna be a rock climber, you probably ought to go overseas, right? Scandinavia, right? If you're gonna do stuff that'll transect your spinal cord. So anyway, that was kind of an interesting one. What?
Safer than driving.
Probably, yeah. Probably is safer than driving. That is still going on. Somebody in my last class asked, is that technology going to come back? And my thought was, not in the next 4 years. I'm not particularly hopeful. What?
That's an alternative fact.
Oh, yes, it's definitely an alternative fact. And it also requires that you believe in science. Believe that the scientific method has anything to offer your life. So, and I do, but apparently I am an alternative now. Okay, so let's briefly start on the next set of notes. We're not going to do anything hard on this one, don't worry. We're basically just going to talk about how the brain protects itself, which seems really, really important after just talking about that, right? Is the brain and the spinal cord protected? So this set of notes goes through the brain and cranial nerves. And of course, you guys remember that the cranial nerves, so the brain is the brain, obviously. The cranial nerves are those 12 pairs of nerves that are the on-ramps and off-ramps directly to the brain. The first lab after your practical, you guys will do the brain and cranial nerves, and you will do a sheep brain dissection. So you will be probably a little ahead of most people on that stuff, which is kind of good that I ended up doing the brain first, because then when you guys are doing your practical, I'll be doing the skin, which is super easy. And you'll be like, ugh, skin's fine, whatever. Instead of the brain, which is like super hard. Okay. So let's talk about the supports and protections around the brain. So the first support is basically this big honkin' skull around the brain, and then like the vertebra around the spinal cord. Now, all of us have fallen on our damn heads, right? But very few of us have cracked our heads open. So that's pretty effective, right? So the cranium is a really, really effective case for your brain. But then there are multiple protections underneath that. The protections underneath that include 3 sets of membranes that are protecting, and they're collectively called the meninges. I'll teach them to you in just a second. And then in addition, your brain has fluid surrounding it on the outside and the inside. That's called cerebrospinal fluid. And all of that is in addition to the thing called the blood-brain barrier. And the blood-brain barrier protects you from whatever's going through your bloodstream. So this is probably helping you understand why almost none of us have ever had a brain infection, right? It's the most badass, multi-tiered protection that we have evolved. So let's talk about the layers of protection below this, so the meninges. So I'm going to show you around the spinal cord, and then I'll sort of draw a bad picture of it around the brain. Okay, so deep to the bone, what you're going to have is 3 layers of membranes that are protecting the brain. The first one is called the dura mater, and it goes all the way around the brain and the spinal cord collectively. And dura mater literally means tough mother, and it is a tough mother. Okay, it is hard to puncture on accident. It is opaque. You can't even see the brain when the dura mater's on there. It is like the thickest of dense, irregular connective tissue. And if you get through it, it's probably not an accident, or else it was a really, really serious injury. Has anybody ever had or seen a spinal tap done? Spinal tap, in order to get the cerebrospinal fluid out, you have to puncture the dura mater. Have you seen that freaking needle? It is a beast. That needle is a beast. It's got little gradations so you can tell how far to go. And usually you have to like get some elbow grease in to actually get it, and you'll hear it pop. So that is not something that you can injure very easily on accident. Of course, you can injure anything if you try hard enough. So that's the dura mater. The next one down, do you see how it's spiderwebby? That one's called the arachnoid or arachnoid mater, the spider mother. So you got your tough mother, your spider mother, and then on the surface of the brain and the spinal cord, it doesn't even look like a separate membrane. It just looks like the surface of the brain and the spinal cord is shiny. That's called the pia mater, and the pia mater means gentle mother. So we have bone, right? We have fluid. We have a tough mother, a spider mother, and a gentle mother, and the blood-brain barrier before you actually get anything directly to the brain. So let's talk about how this works all the way around the brain and the spinal cord. So I'll draw up just a bad picture so that you can actually see that it's all continuous. Okay, so that was around the spinal cord, but what I want you to understand is that it's actually a continuous layer. So imagine that this is not a terrible picture of the brain. Spinal cord. So let's look at the layers in order, okay? The layer that touches the brain and the spinal cord is continuous, all the way through, going up the foramen magnum, all the way around. And it actually touches, I'm drawing it slightly separated, but just so you know it's actually intact, in contact. This is called the pia mater. You can't really get the pia mater off of the brain and the spinal cord very easily. You can if you have some tweezers, but a lot of times the tissue comes off with it. So if you're trying to pull it off with a sheep brain, and you should, just for curiosity, right? You'll see that sometimes the tissue actually comes with it. The next layer up that's superficial to that is going to be the arachnoid, or arachnoid mater. And then the most superficial layer and the thickest one is the dura mater. But then there are some spaces. The spaces are also important. Okay, so dura mater on the outside. There isn't really a space superficial to the dura mater in the cranium because the dura mater is stuck to the periosteum of the bone. Okay, so it doesn't really have a space around there. But the spinal cord, of course, is wigglier, and it has to have a little padding between the bone and the dura mater. So let me show you that. So imagine that that circle is a vertebra, right? Superficial to the dura mater, between that and the bone, the space right here. Do you see it? Or right here. This is called the epidural space. Good? So let's talk about epidural injections for a second. An epidural injection, let's say you were having an epidural injection because you were giving birth and it's freaking painful. Okay, so I know he's not going to give birth. I don't have a female to show you. We don't have to worry about him giving birth. So what we're going to do with an epidural injection is we do not want the anesthetic to go through the dura mater. We want it to say epi to the dura mater, right? And we want to inject it into the fat tissue, right? It's basically got fat tissue, but we want to be specific about where we inject it because we know that the nerves that are leaving places actually innervate tissues that are lower to them. So first off is you want to make sure that you always go below that L2, which is where the spinal cord ends, right? So if you ever made a mistake and damaged nervous tissue, you want to damage PNS, not CNS, right? And then what you want to do is you want to figure out where the roots go that innervate the parts that are going to hurt. Yeah? And then you inject into that area, but it's really not exact. You inject into that general region, and you inject into the fat tissue, and then it diffuses. It actually doesn't deaden the central nervous system tissue because it's got all those protections. It deadens the spinal nerves that are coming out that region, right? That's not very specific because if you lay it on your right side, you get more anesthetic effect on your right side, left side more on your left side. Stick your butt on a pillow, you'll be numb all the way up to here. Yeah? And then a lot of times there's no way to anesthetize one region without, for instance, having paralysis or loss of sensation lower. It's temporary, right? It's just a temporary anesthetic. Now, if you get it too far up, the problem comes is that if you get it too far up, you'll lose your abdominal muscle contraction, and then you have trouble with using your abdominal muscles. But it's really an inexact science.
So then it makes sense that it's only going to affect the mother and it can't enter her bloodstream at all.
No, it doesn't enter
It
the
can't
bloodstream.
at all.
Well, there's no can't. If you hit a blood vessel, it certainly can, but it is very unlikely to do
Very
so.
unlikely if you do it correctly.
Yeah, if you do it correctly, because it's in the interstitial fluid or the extracellular fluid. Yeah? That makes sense? Okay, so that's sort of the basis of epidural injections. Now, if you're having a C-section like I was, and mine was planned C-section, and I have a heart problem, and pain initiates my heart problem, it makes it like go completely wacky. So they wanted to make really sure that I wasn't in pain. They injected opiates directly into my cerebrospinal fluid. It's kind of awesome, honestly. I was like, this is great. My husband's like, can I have some? I'm like, hell no. It's all for me. But it's a little more dangerous, of course. So now let's talk, and we'll get back to the layers of the dura mater next time, but let's just briefly talk about the other spaces that exist. Okay, so there are some other spaces that exist. Okay, so we got the epidural space that's superficial to the dura mater, right? And then this space right here, do you see it? This one, right here. It's called the subdural space. There's a bunch of blood vessels that run in there, so you can, for instance, have a subdural hematoma. Yeah? Okay. And then deep to this, deep to the arachnoid right there. The arachnoid is actually really spiderwebby, so I want to show you what the arachnoid is actually like. The arachnoid is spiderwebby, and the spider webs really connect the top of the arachnoid to the bottom of the pia mater. So this space that's within this region right here, they call it the subarachnoid space. Subarachnoid space. But it's actually, to my mind, it's like the intra-arachnoid space, because that should have been there. That one's really important because cerebrospinal fluid circulates in there through all the, there's a bunch of spaces in the spider webs. So the space that's deep to the arachnoid or within the arachnoid is actually called the subarachnoid space. And that's where cerebrospinal fluid circulates. And then you hit the pia mater and there's no subpial space. That make sense? Okay. I think I have a picture of it here too. Let me show you a figure. I'll just show you the subarachnoid space real quick and then. Not that one. As soon as I find the picture, I'll show it to you. A second. Just want to give you a visual of that one and then I will. Okay. All right, so briefly, and then I will let you guys go because this is as far as the other class got. Do you see where we're looking right here? Okay, so, and we'll look at this in more detail next time. Okay, we've got the skull. This gray space right here, the pia mater is represented in white. Do you see it? This gray space right here is the arachnoid, yeah? And then the dura mater you're seeing in white right there is actually a two-layered one. So what I want to do is blow that up so that you can see what the subarachnoid space looks like. So do you see this? Cerebral cortex, yeah? Pia mater. this is both the arachnoid and the subarachnoid space. Yeah? That's why I wouldn't call it subarachnoid if they let me name it, which they didn't. I'd call it the intra-arachnoid space. And this is important because this is where cerebrospinal fluid circulates. And there's a mechanism of getting the cerebrospinal fluid back into your bloodstream, which we'll talk about next time. Does that make sense? Okay, so we'll go through this in more detail next time. I feel like I'm forgetting to tell you something, but I'll figure it out by next time. Let you guys go early today.